All right, so 9.3 is we're going to talk about mitosis and cytokinesis. So we've already went through the interphase portion of the cell cycle. So remember, some of you didn't do the correct one pager. The cell cycle is that circle, it's a cycle. So it's the circle diagram. She's going to give some of those back to you, and you can redo them for full credit. So first we're going to talk about, remember mitosis is nuclear division. So what's in the nucleus? DNA. So we're going to talk about DNA first. So your chromosome number, or an organism's chromosome number. So when not dividing, your DNA is located within chromatin or relaxed chromosomes. So it's kind of like loose string. It looks like a bunch of tangled thread. So before mitosis, before it gets ready to replicate and split, or I should say split because it's already replicated in the S phase, chromatin becomes condensed and highly coiled, which is called a chromosome. So the two things that you need to remember from this slide are that chromatin is loose DNA and chromosomes are tight DNA that happens right before the nucleus divides. Okay, it's kind of like prepping and getting ready. It's almost like cleaning your room. So you need to underline this in your notes. A diploid number, which is you can write 2N. So N is like the actual number of, or the number of types of chromosomes you have, times 2, which will be the diploid. So this is 2 of each, okay? Like you would have 2 bananas, 2 apples, 2 oranges, things like that. Diploid number is the number found in every cell of an individual or chromosome or organism. It includes 2 chromosomes of each kind. Like I said, it's like if you have a fruit bowl and you had 2 of every fruit. That is diploid, okay? You in your cells have 2 of each type of chromosome. You got 1 from your mother and you got 1 from your father. And then you have genes on those chromosomes that we'll get into later that can dominate over the recessive one, and that's why you have the color eyes you have or the color hair you have, okay? Then you have haploid number, which sounds exactly like what it is. It's half the number of diploid. It contains only 1 chromosome of each kind, and the only cells in an organism's body that have the haploid number is the reproductive cells called gametes, aka the sperm cell and the egg cell. Those are the only ones, because a half and a half make a whole, right? That's haploid. Everything else in your body has 2 of each chromosome. Your diploid number is 46, and your haploid number in your reproductive cells or gametes is 23. All right, so prep for mitosis. Chromosomes duplicate. The dividing cell is the parent cell. Like I talked about before, the resulting cells are called the daughter cells. Then you have sister chromatids that are attached at the centromere. So if you look over there on the diagram, 2 sides, I know it looks like it's an X, but one side of that, the chromosomes don't actually cross, if that makes sense, like an X. They just touch in the middle at the centromere. So you can see that each side of that orange X over here, that chromosome, each side is a chromatid. So those are sister chromatids right there. Then you have these kentochores, which develop on either side of the centromere. So they're almost like a handle for the microtubules that are about to pull it apart. Those are the pink ovals you see. You need to know both of those for your test. So that was the chromosome duplication. This is the division of the centrosomes. So centrosomes was part of, I think it was chapter 3 or 4, when we learned about all the organelles and the cell parts. Well, centrosome is a cell part. You can see the little bitty microtubules lined up. Those are just it looks like a spaghetti noodle. The centrosome is a microtubule organizing center. It divides before mitosis. They contain two barrel-like centrioles. These are not found in plants. They organize the meiotic spindle. Microtubules are hollow cylinders made up of tubulin. That's another kind of like definition down here at the bottom. So centrosome is two centrioles. Okay, these are centrioles. This is a centrosome when you have both of them together. And this is where the microtubules come out, almost like growing fingers, and attach to those pink kintochores right there and pull those apart. All right, so we're going to go over the phases of mitosis. And this is what some of you drew, I believe, for your one-pager accidentally in place of the cell cycle. You have to know the difference between these two. And that's why I'm allowing, instead of just giving you a 0, I'm allowing you to do it over because I really want you to know the difference between the cell cycle and mitosis. Mitosis is part of the cell cycle. Where's my picture at? Hold on. Let me find another one real quick so you can see. Google does this thing where like you can't, it only does images that you can like actually use. I won't just steal an image. It's pretty good. Let's do this one. Oh, I misspelled that. Okay, that's a little bit better anyway. So prophase is where chromatin condenses and the chromosomes become visible, because remember, chromatin is just that, it's like your messy room, and chromosomes is when you tidy it all up. So the chromosomes are condensed DNA, okay? Duplicated chromosomes already, remember that from the S phase. So the nucleus disappears, the nucleolus disappears, and nuclear envelope starts to disappear too, and the spindle assembles. So I will ask you what happens in prophase. I've always taught it to where there's 3. I need to take this nucleolus off because I know that sometimes can confuse you. So the 3 things you need to know about prophase. Number 1, chromosomes become visible. Number 1, chromosomes become visible. Number 2, the nuclear envelope dissolves. Or you can put NE if you know what you're talking about. That's that little pink. This is the nucleus right here. It's starting to break apart and, as your book says, fragment. And number 3, the spindle assembles. So you see these little centrosomes right here in the purple. These microtubules are starting to grow fingers, be little finger growers, I guess, and get ready to start attaching to these little kintochores. Okay, so 3 things. Chromosomes become visible, the NE dissolves, and spindle forms. Okay, 3 things. Prophase is the prep phase. We're not going to go over pro-metaphase or late prophase because we're just not going to. But this is another picture of almost, essentially prophase. It's really good. You can see the nuclear envelope dissolves. You can see the chromosomes have become visible. They've condensed. And you can see the centrosomes have are making microtubules, which is essentially called the spindle. They're about to get these lined up and attach. So don't copy this slide. All right, so the next phase in mitosis, or the way I like to remember it, is PMAT. So P stands for prophase, M stands for metaphase, okay? Metaphase is where the chromosomes line up in the middle. Metaphase, middle. Or as scientists call it, the metaphase plate. I'll take either one. You can say chromosomes are lined up in the middle, or you can say chromosomes are lined up at the metaphase plate. And that's literally all that happens. You see these chromosomes that condensed in prophase line up in the center of the nucleus. You see these spindles are attaching to those kintochores right there, and they're about to pull them apart. So in PMAT, P stands for prophase, M stands for metaphase, and A stands for anaphase. Okay, anaphase is where you're going to pull those chromosomes apart. So chromosomes separate into two sister chromatids. They're being pulled apart by the spindle. And this is the shortest phase of mitosis. So literally the only thing you need to know is anaphase is where chromosomes are pulled apart. That's it. So again, I'm trying to make you remember it. Prophase is prepping, getting ready for it to divide. Chromosomes become visible. The nuclear envelope dissolves and the spindle forms. Then in metaphase, the chromosomes line up at the metaphase plate, or the middle. Anaphase is where the chromosomes pull apart into sister chromatids. And the last part of PMAT is T, which stands for telophase. So think about a party. Like if you ever had I don't think y'all even do this anymore. I know Miss Willis and I did. We used to have co-op parties. You could rent out like a little portion of the co-op, which is so weird, I guess, for events. And we would have our birthday parties there and like have dance. It was so cheesy, but we would have like dance parties and stuff. And so before everybody got there, we would all come and set out chairs and, put the cake out, and we would put streamers out and get ready for the party. Like, think about putting out fold-out chairs. You fold them out. That's prophase. Well, then the party happens, which is metaphase and anaphase. And then after everybody leaves that party, what do me and Miss Willis have to do to those fold-out chairs? We have to fold them back, right? You just do the opposite of what you did before. That's telophase. So guess what? The spindle disappears, the nuclear envelope forms back, and the chromosomes, you can say dissolve, you can say uncoil, you can say whatever you want, but chromosomes turn back into chromatin, that loose chromatin. So literally the three things that we learned from prophase happen in telophase. It's just the opposite, okay? Just like when you have to clean up after a party. During telophase, you can see that the nucleus is forming now on each side, and both of them have the exact number of chromatids as the other side. Who checks that for us? That G1 checkpoint, remember? So lastly, cytokinesis is some people include it in mitosis, some people don't. It's just the division of the cytoplasm. If this doesn't occur, because sometimes it doesn't, you can see that under a microscope sometimes, cells become multinucleated. They just have two nuclei. So cytokinesis kind of begins in anaphase, but doesn't complete until the following interphase, which is weird. So let's look back at anaphase. So you can see metaphase, it just looks like one big blob, right? Well, look in anaphase. Metaphase, you didn't see a little hump, but right here you can see that there's two cells starting to form. And you can see in telophase it's almost finished. So that's cytokinesis, the division of the cytoplasm. It's a little different for animals and plants because they have, one of them has a cell wall. I'll leave that to you to figure that out. I'll say it maybe in a minute, but you should know that already. So in animals, there's a cleavage furrow that happens. I will ask you this on the test. A cleavage furrow is an indentation of the membrane between the two daughter nuclei. It deepens, it's almost like a drawstring happens with these little filaments called a contractile ring. It just like cinches up like a pair of sweatpants and in between them until they pinch off like Play-Doh. That's in animals. You can have Miss Willis stop me if you need to. And in plants, you have to build a new cell wall. So guess which one has a cell wall? Plants. So this is an actual picture of a plant cell, by the way. So the Golgi apparatus in plant cells produce vesicles. Remember, vesicles are just these little taxis that fuse in the middle of the cell. You can see this line starting. They just make a bunch of vesicles and they line them up in the middle. And they form a cell plate. That's what that pink line is right here between these two nuclei. This cell plate forms into a newly plasma membrane, and eventually the cells will add enough fiber and cellulose and things like that to make the cell wall, and it'll become a cell wall itself. So plant cells have a cell plate. The Golgi produces that. In animals, they have a cleavage furrow, which is just almost like if you take scissors and snip something and you pull it apart, or a drawstring in pants. You can't do that with plant cells, though, because the cell wall is so rigid. You can't cinch it. You just can't pull it apart. We're getting close to being finished. Closer. So the functions of mitosis. We have mitosis that permits growth and repair. If you scratch your arm in a few days, you probably won't be able to tell it, and that's because of mitosis. Our skin cells have recreated those cells and repaired our cut. It's also necessary for an embryo. Just think, we all looked like this little guy down here at some point. All right, 9.4. So we're going to talk about cancer next. So cancer is cellular growth. It's a cellular growth disorder that occurs when a cell divides uncontrollably. It's just uncontrolled cell growth. That's all cancer is. It's when those checkpoints kind of become mutated and we lose control of the cell cycle. Is that not crazy that we think of cancer as this like crazy thing that we don't understand because we're not doctors? Well, you really can understand it because all it is this chapter. If you know this chapter, then you know what cancer is, because those checkpoints in the cell cycle, either the cells have mutated and they're not there anymore, or maybe, mishap in your DNA and they didn't recreate for the next cell. So it's like the cell cycle going 120 miles per hour all the time. That's what cancer is. You have two types. You have benign cancer, which is non-cancerous. These you'll hear like a benign tumor. That means it's not growing. It's not currently going 120 miles per hour. So maybe your cells, maybe your body stopped it at that point. Then you have malignant. Malignant means it's actually cancerous or spreading. Okay, it's getting bigger, and what happens with cancer is it can spread through your blood and travel throughout your body. Your blood system is like the highway of your body. And if those cancer cells hop on one of your little blood cell inner tubes and travel down, it's almost like a lazy river too, and travel to a different part of your body and get off, then that part of your body also now has cancer, and that's when it gets bad. We've all probably heard, the cancer has spread. That's how it spreads, through the blood. So a cell that has a mutation may take decades to become cancerous. So I think about it all the time. I'm at home right now, all I do is eat junk food. So as terrible as it sounds, I'm probably just putting cancer food in my body, because I'm not eating right at the moment. So, I probably have cancerous cells in my body right now. You could have cancerous cells in your body right now. Who knows? So the characteristics of cancer, usually the cells will lack differentiation. So cells have certain jobs in the body, right? So, and they all look different based off of what their job is in the body. So these cancer cells are kind of like, it's like they don't care about what their actual job is. Their job now is they've been hijacked, and all their job is now is to just grow and divide. Most of the time, their nucleus looks weird. It looks abnormal. Apoptosis does not occur for cancerous cells, and they form tumors. And they can undergo two different things, okay? So you need to know these two things. So metastasis is when cancer cells travel somewhere else. That's like what I just talked about. And angiogenesis is when there's a tumor, and your blood cells will actually form around that tumor and feed it. That can be very bad, because it's getting all those cancer cells are getting direct nutrients from your blood. That's angiogenesis. Trying to think of a way you can keep those two apart. Let's see. I guess you'll just have to learn them. I can't think of any silly analogy. So metastasis and angiogenesis, those will be on your test. So this is just a picture of cancer. So at the very beginning of this picture, you can see this tiny little cell over here has a mutation. Well, later on down the road, it gets a little bit bigger and it starts growing and growing. And then this picture right here with this purple in it is cancer, okay? And then you have what's called invasive cancer, and this is where metastasis happens. You can see the cancer has gotten huge, and it gets in the bloodstream, and it travels elsewhere. So the origin of cancer, there's two genes that are usually affected. There's proto-oncogenes, These promote progression through the cell cycle. When there's a mutation, this oncogene, this is a cancer-causing gene. It almost forms, and that's what the mutation is, okay? And then you have tumor suppressor genes. Inhibit the cell cycle and prevent uncontrollable dividing. So a mutation in this is like a brake failure in a car. So I was talking about going 120 miles per hour. That's the gene that's one of the ways cancer can work. All right, this is our last. Let's see how many we got left. We're almost done. So the last thing we're going to talk about is prokaryotic cell division. So prokaryotic cell division, you have asexual reproduction. Prokaryotes don't produce sexually. Sexually means there's two people involved or two organisms involved. There I go again. I always talk about just people. Asexual reproduction is when offspring are genetically identical to the parent. The prokaryotic chromosome, remember we lack a nucleus and other membrane-bound organelles. And we have one single chromosome called the nucleoid. If you stretch that nucleoid out, it would be a circle. All right, so binary fission. So I like to think of the word fission. The opposite of fission is fusion. So when you think of fusion, what do you think about? Two things fusing together, right? So fission is pulling apart, basically. So this is the way prokaryotes reproduce, one of the ways. Division produces daughter cells that are identical to their original parent. Elongation of this cell that pulls the cell apart. So it just stretches out, and then a plasma membrane and cell wall form between the two cells. That's all that happens. Super simple. Pi means two, right? So you're pulling it apart into two cells. So here's our picture of a bacterial cell. There's one nucleoid, right? It would be a circle if we pulled it apart. There's no nucleus, so we don't have to have mitosis. We don't have to break down the nuclear envelope and things like that. We do duplicate our chromosome and separate the copies. We stretch out or elongate, and then we just divide the two cells. That's binary fission. I will ask you what binary fission is. So the last two slides we have are comparing prokaryotes and eukaryotes. Remember, eukaryotic cells have a nucleus and prokaryotic cells do not. So remember, prokaryotes are single-celled. There's literally one cell per organism. They use a form of sexual reproduction because I don't know why I put on. Maybe I meant one. I'll do this next year too. I'll be like, why did I put on? Because one parent produced an identical offspring. And there's no spindle in binary fission because there's nothing to really pull apart. There's just one, right? That's how prokaryotes divide. Eukaryotes, cell division is a growth process. It's not a type of sexual reproduction. So that's what you're going to try not to get confused on, okay? Mitosis is just used for growth and repair. Remember that slide? We just grow. Like that plant on the left just grew because of mitosis. That cat grew that big because of mitosis. There's a whole nother chapter for our type of sexual reproduction and what our cells go through. So we use it to grow. Prokaryotes use it to divide. Okay? And that's it.